Hello everyone. Hope everyone is fit and fine. Welcome back to Shine. In order to shine, today we'll be learning a new topic, that is membrane transport under the topic cell biology. This is part 2. In part 1, we have discussed about the cell and its components with their different types of functions. In this part, we'll be learning about the membrane transport mechanism. Like in previous slides, we have already discussed about the membrane, cell membrane, which is made up of phospholipids bilayer and which acts as a barrier between the internal and the external environment of the cell. So in order a molecule wants to get inside or any substance wants to come out of the cell, it has to undergo by certain kinds of transport mechanisms. Now let's see them in details. Under the membrane transport mechanisms, we have three different types of transport mechanisms. The first one is transcellular transport, second one is paracellular transport, and the third one is vascular transport. Transcellular transport is defined as the passage of the drug or the molecules across the GI epithelial cell. This transcellular transport gets divided into three types, that is simple diffusion, fasciculated diffusion, and the active transport. Now this active transport gets divided into two types, that is primary active transport and the secondary active transport. The second type of transport is known as the paracellular transport. In this transport, the molecule is transported between, or we can say it passes between the junctions of the GI epithelial cell. Now the last one but not the least is the vascular transport. In this, the transportation takes place with the help of energy between the cell, or we can say inside the cell by the formation of the vesicles. It is of two types, that is endocytosis and the exocytosis. Now let's see each mechanism in details. The first one under the transcellular transport is the simple diffusion. This diffusion is also known as passive diffusion. It is known as simple diffusion because the transportation of the molecules is taking place from higher concentration to lower concentration. And in this type of diffusion, the size of the molecules are small, means the size of the molecule is less than 500 Daltons and the molecules are non-charged, or we can say uncharged. Since the movement is taking place from higher concentration to lower concentration, it does not require ATPs or the energies. So this type of movement, which is ATP independent, is known as or we can say it is called as the downhill movement. The calculation, or we can say the rate of diffusion, can be calculated by the help of Fick's law, or by this formula, that is surface area into concentration gradient divided by thickness of the membrane into weight of the molecules. Now let's see the examples of the certain molecules which get transported with the help of this diffusion are respiratory gases like oxygen or the carbon dioxide, certain hormones like steroid hormone or drugs which are lipid soluble, they passes easily with the help of this type of diffusion. Now the next one we have is fasciculated diffusion. Fasciculated diffusion is also called as the combination of the passive diffusion and the active transport. Now, why it is called as the combination of the active transport and the passive diffusion? Because like passive diffusion, the movement of the molecules is taking place from higher concentration to lower concentration. But the size of the molecule is large, means the size of the molecule is more than 500 daltons, and molecules are charged. Since the molecules are charged, they requires a carrier like active transport. That's why it is known as the fusion or the combination of both the passive and the active transports. But it does not require ATPs like active transport. Now the certain carriers which helps the facilitated diffusion are aquaporins, leaky channels, voltage gated channel, ligand gated channel, mechanically gated channel, and the glute transporters. Now let's see each carrier in details. This is the diagrammatic representation of all the different carriers which help in the facilitated diffusion, like aquaporins, leaky channel, voltage gated channel, ligand gated channel, and mechanically gated channel, and the last one, glute transporters. The first one is aquaporins. They are also called as a plumbing system of the cells. They are consist of 6 transmembrane alpha helix which are embedded within the cell membrane. See here, aquaporins. They are mainly responsible for the transportation of the water, but they depend upon the concentration of the solute. Means where the concentration of the solute is high, the water flows in that direction, from that direction. Now the next one is leaky channels. Leaky channels are called leaky channels because they are always open and they are potassium selective. They control the resting membrane potential of the neurons. See here. The potassium, which is present in higher concentration within the cell, moves from inside of the cell to outside of the cell with the help of these leaky channels in neurons. Now the next one we have is the voltage gated channel. When a threshold reaches to 55, this channel gets opened up and transportation of the sodium from outside environment to inside environment of the cell takes place. The sodium is present in outside the cell in higher concentration, and it mainly affects the neuron. Now the next one is ligand gated channels. When a chemical comes and sits on the pocket, see, when a chemical, that is acetylcholine, comes and sits on the pocket, this pocket or this channel gets opened up and the movement of sodium from outside environment to inside environment takes place. It happens, or it controls the neuromuscular junction. Now the next one is the pain receptors, or we can say the mechanically gated channel. When a mechanical force is applied, the pain receptor or the channel gets opened up. Means a mechanical force is applied, the sensory nerves get stimulated with the signal, and the signal is sent to the CNS to open up the channel for the movement of the sodium inside the cell. Now the last one is the glute transporters. They are mainly responsible for the transportation of the glucose by the help of downhill movement. Now the next type of transport is active transport. This is called as active transport because they are utilizing the ATPs, means energy for the transportation of the molecules. Here the molecules are getting transported from lower concentration to higher concentration. For example, like when we travel or when we climb a mountain, we require more energy to climb it up than to climb it down. or to climb it up and to come down. So same thing happens here. When a molecule is getting transported from lower concentration to higher concentration, it requires an energy for its transportation. This active transport is divided into two types, that is primary active transport and the secondary active transport. The main difference between these two is that primary active transport uses the ATPs directly, whereas secondary active transport uses the ATPs indirectly. Now let's see them in detail. In primary active transport, whatever the movement takes place or the transfer takes place, it happens in one direction. That's why this type of transport is known as uniporters. Here the transport of only one ion or one molecule takes place, and the molecules which are transporting, they are larger in size and they have charges present on them. See here, in examples you can see the sodium potassium ATPase. Here the exchange of sodium and potassium ion is taking place. Only one ion is coming in and the one is coming out. See, the next one is sarcoplasmic reticulum, in which the calcium concentration is more. Means from the inside environment, the calcium is going to sarcoplasmic reticulum. Means from low concentration to high concentration is going. The next example is the H plus ion and the potassium ATPase. Here in the stomach, the H plus ion gets secreted and it makes the environment acidified. So when we give the drug to deacidify, mainly the proton pump inhibitors, they come and they bind on this particular region to deacidify the environment. Now the next type of transport is secondary active transporters. In this secondary active transport, two types of transportation is taking place, that is symport and the antiporters. Sympoters means the molecules are moving in the same direction, whereas in antiporters both the molecules are moving in the opposite directions. Here you can see the example of symporter is the sodium and the glucose. They are moving in the same direction in the kidney tubular cells. Or you can also see in the loop of Henle in kidney the transport of sodium, potassium and the chloride ions. But here when sodium or we can say the sodium and the H plus ion, here by the help of the concentration gradient of the sodium, the H plus ion is getting excreted, or we can say it's coming out. So they are antiporters, they are moving in opposite direction in the density of the kidney. Now let's see the next type of transport mechanism, that is the vascular transport. Under vascular transport we have endocytosis and exocytosis process. Here endocytosis process, here a molecule to get absorbed does not need to be in aqueous state or in an aqueous solution. In endocytosis process, it mainly uses three mechanisms, that is pinocytosis, phagocytosis and the receptor mediated endocytosis. This endocytosis process, when whatever the transportation is taking place of the molecule, it is happening within the vesicle or within the cell. Means the vesicle is getting formed and the transportation is taking place within the cell. Let's see how it's happening. This is the diagrammatic representation of all the three processes which help in the endocytosis process. Pinocytosis, also known as the cell drinking process or the cell drinkers. What happens here? The microtubules with the motor proteins, they move the pinocytic vesicles inside the cell. See, the microtubule with the motor protein is coming and it is pushing the pinocytic vesicle inside the cell with the help of ATPs. Next one is phagocytosis. Here the engulfment is happening. Mainly in endocytosis, engulfment of the material takes place. When a bacteria or the pathogen enters, the H plus ion gets activated, and with the help of lysosomes, with the help of H plus ion, lysosomes get fused with the phagosomes and does the lysis of the bacterial cell, and that waste material is then left in the cell for the further process. And the last one is the receptor mediated transport. Here the H plus ion moves inside the endosomes and it divides the vesicles into two types, the one which contains the receptor and the other one which contains the low density lipoproteins. Now the lysosomes gets combined with the low density lipoproteins and leaves it breaks down the molecule into small fragments for the further process inside the cell. Now let's see the last process or the mechanism for the transportation, that is exocytosis. The main difference between the endocytosis and the exocytosis is that the exocytosis process throws out the waste material outside the cytoplasm, or we can say from whatever the waste material is left in the cytoplasm is transported outside to the cell excretory parts. See here, from DNA after transcription gets converted to RNA, and then from translation protein is formed. Protein is modified by Golgi apparatus and it is utilized. When the utilization has been done, whatever the material or the waste part is remaining is being thrown out of the cell by the help of this exocytosis process. Up to this, now let's have a recap. In this part, we mainly discussed about the mechanism of the transportation of the molecules from the cell. There are mainly three types of mechanism, that is transcellular, paracellular and the vesicular. The trans is divided into three types, that is passive, facetylated and the active transporters, in which passive diffusion and facetylated diffusion does not require ATPs, and facetylated diffusion is known as the combination of the active and the passive diffusion. The active transporters are of two types, that is primary and the secondary active transport. Last one is the vascular transport. Under vascular, there are two types, endocytosis and the exocytosis. Endocytosis is the process where compound does not have to be in aqueous solution to get absorbed, whereas in exocytosis, the waste material is expelled out of the cell. It does not remain inside the cell, unlikely like endocytosis process. Thank you. If you have any doubts or you want any material regarding to this topic or the previous topic, you can contact me on this Instagram ID, that is shine2109, or you can comment down your doubts in the comment section. Do like, subscribe and share this video if you want more educational video to make you shine among everyone. Thank you.